All right, maybe I'll get started. It's a great pleasure on my part to introduce our speaker today, Chi Van Dong. His story should be very inspirational. He came to this country as a young boy from Vietnam in 1967. He went on to get a bachelor's and PhD in chemistry, and then also an MD in 1982 from Johns Hopkins. He did intern residency at Hopkins, and then took a fellowship at UCSF during the period from 1985 to 1987. It's during that time that he first started working on MYC, the oncogene. He then returned to Hopkins as an assistant professor and rose through the ranks, with all sorts of positions and titles, became a full professor. And then in 2017, became the scientific director of the Ludwig Institute for Cancer Research, which is a large network consisting, I think, of 4 places internationally, and then 6 domestic research centers around the U.S. His own lab is in the Wistar Institute in Philadelphia, and he's been director of this network for quite some time, since 2017. As you would expect from that kind of intro, he's had a lot of awards. He's a member of the National Academy of Medicine, American Association of Arts and Sciences. He's the chair of the National Cancer Institute's Board of Scientific Advisors. He's advisor to the president about cancer-related issues. And all of this has occurred because of the work in his lab focused on MYC and their major, and they've published on many, many aspects of MYC. But the one he's going to focus on today is its role and link to metabolic change, and how tumorigenesis results in sort of a change in the metabolic program, and how circadian rhythms play into that. So with that, Chi.
Great. Well, thank you very much, Jerry. I really appreciate the invitation to be part of this spring colloquium, and always enthusiastic to look after this to meet with the students. So as Jerry indicated, maybe one note in a very nice introduction is that just for the students, I think that one thing that I've learned is that luck and timing is extremely important, because when I was a postdoc at UCSF, among many oncogenes, I happened to choose MYC to study under Mike Bishop and Harold Varmus. And it turns out that, as Jerry indicated, picking this gene actually have paid a lot of my bills over the last 30 years. So that was a pure luck. So let me just start by telling you the topics we're going to cover today. I'm going to start out by distinguishing between maintenance metabolism and what I term as proliferative metabolism. That is, when cells are stimulated to grow, what's the difference between these two types of metabolism? And then I'll turn a little bit into our work, and just which has already been published, to talk a little bit about this role of MYC in the circadian clock, and touch briefly on the work that we did in Drosophila MYC, and what we found there in terms of loss and gain of function, as well as implications on disruption of metabolome that might affect circadian behavior of fruit flies. Then I'm going to turn to a topic that is, much more of a key interest in my lab, is that to look at how MYC drives tumorigenesis and metabolism. This is where the connection between MYC and the clock was uncovered by us about half a decade ago. And I'm going to just show you some more recent studies we've done and then turn a little bit into, because of my background and wanting to translate things, whether our understanding of this could lead to any kind of insight that allows for targeting cancer metabolism for therapeutic purposes. So as you all know, with your very basic biochemistry, that the metabolic map, which is shown on the left, is I would say quite challenging, because none of us can really remember all the metabolic pathways. But I think that if you step back and look at the organization of how metabolism is organized, you'll find that the central path here that I colored in is the energetic pathway, which is that cells take in nutrients. The primary nutrients that we know about in humans is, of course, glucose, which is highly regulated by the insulin system. And once glucose enters the cells, it undergoes glycolysis, and then pyruvate then enters a TCA cycle, which generates a vast amount of ATP as compared to glycolysis. But then if you look at the organization on the left, actually these complicated pathways have organization to it. That is, the central input of nutrients, in fact, can spin to the side to create complex carbohydrates, or for nucleotide metabolism, or a generation of amino acids and the formation of lipids, for example. So in essence, a cell that's not growing will take on nutrients to try to maintain this so-called maintenance metabolism, so it can use biosynthesis when it needs it to repair damaged organelles or proteins that have turned over, that you have to make new proteins. And it turns out, if you look at the total ATP levels in cells, 60% of it actually goes towards membrane potential maintenance, as well as ATP for protein synthesis. So clearly these are required. And then I already alluded to the fact that nutrients come into the cells, illustrated on the right here, actually can generate various components of macromolecules that can generate nucleotides, proteins, and lipids. It turns out that maintenance metabolism is quite strongly regulated by the circadian clock. And what's shown here is that we know that we have adapted, and many organisms actually have adapted to the solar cycle of day and night. And it is believed that we have a clock centrally, which I'll describe later, that synchronizes all the peripheral clocks in your body, so that individual cells have this cycle of activity and rest that is coordinated with our feeding and fasting cycle to optimize the ability to utilize nutrients, and at the same time during the fasting state to allow cells to repair themselves, or a repair phase. So you may or may not know that the central transcription factor in mammalian cells is Colloquium B-mal, and I'll describe the circuitry later, but this regulates a circadian oscillatory transcription of many metabolic genes. And the bottom right is just a cartoon to just illustrate that studies from mouse livers collected at various times of the day, if you look at these, the transcriptome of the liver, as I will also illustrate later, that it oscillates quite dramatically. And then if you look at the actual metabolic pathways that are regulated, the protein levels for some of these enzymes also oscillates. And then what I show here in this outside circle is, if you look at the metabolites, they also oscillate in a circadian fashion, so that you can see that there are various cellular processes that are segregated between night and day for mice, which they're more active at night times, accompanying mitochondrial fission, protein synthesis, as well as unfolded protein response pathway. And then at the resting phase, you'll see more activity in terms of mitochondrial fusion for repair of damaged mitochondria. And you can also see DNA repair being heightened during the resting phase. So the circadian clock, as I already referred to before, is comprised in mammalian cells of Colloquium B-mal. They bind to this hexamer consensus sequence on DNA called the E-box. And it turns out that this heterodimeric transcription factor regulates two negative feedback loops. They activate expression of Cry and PER proteins, which feeds back and regulate, negatively downregulate clock and BMAL in a post-transcriptional fashion, whereas clock and BMAL can also activate an ancillary feedback loop involving the reverb proteins. In this case, reverb alpha and beta transcriptionally shut off BMAL. So you have two negative feedback loops, one that works post-transcriptionally and one is a transcriptional loop. And as you can imagine, in negative feedback loops, this creates an oscillatory levels of BMAL and also activity of clock and BMAL such that the oscillation of activity when they binds to E boxes would drive target genes in an oscillatory fashion in a 24-hour cycle. And just to illustrate this, studies in mouse livers, when you collect it out at various time points, night, day, and in this case, I just illustrate the components of the electron transport chain of the mitochondrion. If you look at the transcript of each of these components encoded in the nucleus, you'll see that the transcript oscillates very nicely in a 24-hour cycle. So we all have a central clock that senses light through the retina, which sends neural signal to the hypothalamus. And this is the central clock sits in the suprachiasmatic nucleus, which then signals to the pituitary gland to release cortisol early in the morning, as shown below here. And that allows for the synchronization of peripheral clocks, which is found in virtually all of our nucleated cells in all different organs shown here. That's been very well documented. And just to illustrate that at the organismal level, as night falls, you can see that the cortisol decreases and then the body repairs itself for the resting phase with an increase in melatonin, which also oscillates in a circadian fashion. Now, a number of years ago, Aleem Welji and John Hoganesh, in part collaboration with my lab, undertook a very interesting studies where we seek to determine the circadian metabolome in the liver. So in this case, at every time point, every 1 hour for 48 hours, 4 animals at every hour is sacrificed, and then the livers were harvested, and then the metabolites were then analyzed by mass spectrometry. And what you can see here in the heat map is that maintenance metabolism, the liver's not growing, is very dynamic. And you can see that many metabolites, in fact, oscillates in a circadian fashion, red being high and green being low. And it's just to illustrate, just picking out 3 metabolites from this map. You can see serine, glycine, and creatine can oscillate very nicely as illustrated here. Now, I want to distinguish this maintenance metabolism, where the cells have to take in nutrients to repair itself in an oscillatory fashion, and compare that to proliferative metabolism. And this is just a cartoon of a cell with a growth factor receptor, whether this growth factor receptor in a normal cell that engages growth factor, or a oncogenic growth factor receptor that can also turn on canonical signal transduction pathways such as RAS and MEK, which we know is upstream of MYC, where it can activate MYC expression as well as stabilizing the protein. And then this allows for MYC and MAX to bind to target genes to produce messenger RNAs, which then, along with mTOR, which is downstream of PI3 kinase and AKT, to amplify this growth signal. So you have the transcriptional amplification followed by a translational amplification. The mTOR signals the cells to build the translational machinery, allowing the cells to build various components, including ribosomes and nutrient transporters, to bring in more nutrients so that the cells not to begin to assemble macromolecules for increase in cell size, preparing the cells then to divide after DNA replication. So this is proliferative metabolism. And just to highlight that we study quite a bit in my lab proliferative metabolism in cancer cells, but it's been documented a decade ago that in normal cells that are activated, in this case, T cells that are activated to their T cell receptor using antibodies, and this is work that was done by Ronan Wang and Doug Green's lab. And what they show in this paper was quite unique in the sense that what they've done is to use T cells from mice that have flox allele of MYC, so you can delete MYC, or you look at flux allele of hypoxia-inducible factor 1, HIF1. And what they showed in this paper is that when you stimulate these cells with the TCR for them to begin to proliferate, that there is a program that we found in malignant cells that completely recapitulate in these normal cells, where MYC activate all the genes that involve, shown here in the blue, in both glucose metabolism and glutamine metabolism. And so when you flux out MYC, you delete MYC out, the entire system shuts down. The cells can't undergo metabolism and they won't proliferate. Whereas if you delete the hypoxia-inducible transcription factor, these cells continue to proliferate in a MYC-dependent fashion. So this is very telling, because the point, take a point home here, is that normal cells use very similar pathways to cancer cells. And I'll get back to this point later. Now, so our interest in MYC has gone on now for a third decade. And this is just to emphasize the fact that MYC is either itself is an oncogene through gene amplification, which is shown here in the red bars here, frequently amplified in many different human cancers from the TCGA initiative. And then MYC is also downstream of various oncogenic transcription factor, oncogenic oncogenes such as mutated RAS, where RAS drives MYC stabilization. And then MYC itself dimerized with its partner protein MAX, and it also recruits a number of cofactors to regulate transcription across the genome. And this is just a crystal structure, a cartoon of the crystal structure that has been solved for MYC and MAX sitting on the E box, being a basic helix loop helix leucine zipper transcription factor. So a few years ago, we were wondering, just looking at the literature, is that if you look at maintenance metabolism or homeostasis, that many of the metabolic pathways are regulated by transcription factors that are very specific, such as ChREPP that regulates carbohydrate metabolism, the FAME SRBP transcription factor that regulates fatty lipid metabolism. But then we focus on the fact that most of these transcription factors bind to short sequences that looks very similar to the E-box. And we noticed that clock and BMAL binds to the hexamer CACTG, which is identical to the MYC E-box. So this is the point where we hypothesized perhaps when MYC is activated, that it could take over maintenance metabolism by activating metabolic genes that would normally be regulated by other transcription factor in the proliferative metabolism. So that was a hypothesis. So let me just turn now, segue into our studies where we asked the question, could MYC itself in a model organism play a role in circadian behavior? And this is work that we've done in collaboration with Amita Segal at Penn, performed by a former graduate student, Annie Shea. So Drosophila actually have MYC and MAX very similar to mammalian cells. They have also cofactors, also very similar to mammalian cells. And it turns out that Bob Eisenman's lab have really generated a series of lines that are very useful for our questions. So basically, what we know is that decreased MYC function, Drosophila MYC function, is associated with small cell size and also small body size, falling into a larger group of flies called minutes. And it turns out there's a naturally occurring minute that happens to be a mutant of DMYC. And so one can study flies that are essentially wild type, or a double mutant where we have a hypomorph of MYC or a deletion of MYC, and the heterozygous that actually creates a MYC mutant that can survive, so one can study where it lacks MYC function. And then it turns out that you can also use another genotype, and I'll get into that in a moment, where there is an antagonist of MYC called MINT. So if you get rid of MYC, you have an unantagonized MINT that shuts off the transcription of what MYC would drive, but you can also delete MYC, MINT to recover the transcriptional machinery that would otherwise be driven by MEC. And I'll show you that momentarily. And it turns out that the flies were where the clock machinery was very well mapped out. Even though the proteins are different, the circuitry is similar to mammalian system that I described before. I won't get into the details of it here, but I just want to move on to show you that we first asked the question in terms of loss of function of MEC, what would happen to Drosophila behavior? And so what Annie Shea did in collaboration with Amita Segal is that we can take flies with various genotype, put them into glass tubes, and then in Amita's lab, they have a setup where infrared beams can detect the activity of these flies. Every time they fly across the beam, you get a tick in the mark called in the actigram. So you can follow the activity of these flies, and they turn out to be very similar to humans, where they're very active during the day, and then they undergo sort of a sleep phase at nighttime, as shown here. So what we did in this experiment is to explore essentially a wild type background and a severe hypomorph, where we have a deletion mutant and also a hypomorph allele in this compound heterozygote. And then finally, what we've done here is also to eliminate the antagonist of MEC called MINT. So the cartoon over here, just to illustrate what we know about MEC and MINT. So MEC and MAX binds to E-boxes to drive transcription. And we know from Bob Eisenman's lab is that MINT and MAX, when they bind to E-boxes, antagonizes MEC and shut off transcription. So that you can imagine if you have loss of function of MEC, which then allow MINT to shut off transcription. Now, if you eliminate MINT, you now can reverse, you can rescue the loss of MEC phenotype, essentially. So what the actogram shown here, and we've done, this is just an example of a single flies, but we've done this with dozens of flies, and the phenotype holds up across all the flies, is that I've compared a wild type. When you have a MEC hypomorph, you can see that the circadian behavior is completely disrupted, notably that the activity during the day is quite diminished. And then these flies also don't sleep at night. They tend to have low levels of activity that is out of sync with day and night. But then this can be largely rescued when we get rid of MINT at the same time. So this triple MINT. And this illustrates to us that this must be related to MYC-mediated transcription. This is what the conclusion, although because we can't study individual cells, it's only by inference to the genetics. And then what we did was to try to glean some mechanistic insights on why there's a disruption of this circadian behavior. And the best we can really do is we harvest whole heads and then subject the heads of these flies with different genotypes to metabolomics. What we find interestingly is that if you follow a wild type, this is a heat map of metabolites, so wild type shown in this column, followed by the mutant MYC, and then also you rescue it with a deletion of DMYT. And we look for metabolites that goes in directions that one might expect, which is it will be high and then low, and then it is rescued when you delete DMYT. And what we find interesting is that histidine is one of the metabolite that behave this way, that also in the gain of function experiment, which I'll go into a minute, where if you overexpress DMYT in certain neurons in the head, you can also see a whole head increase in histidine. So in essence, it's a reciprocal. When you have too much MEC, you have high histidine levels. If you don't have enough MEC function, you lose histidine. So this is one of the metabolite that really goes in the direction of MEC gain or loss of function. And so we surmise and have no real direct evidence at all that perhaps histidine may play a role in this disruption of circadian behavior, because it's known that histidine can be converted to histamine, which does play a role in fly sleep behavior. Because in this paper here, where it's illustrated, if you were to look at mutants of histidine decarboxylase that converts histidine to histamine, that the loss of function of histidine decarboxylate leads to flies that tend to have very prolonged sleep duration during the day, reminiscent of a decreased activity of our MYC hypomorph flies shown down here. So that's just a best guess of the mechanism. And then reciprocally, what we found is that you can overexpress MYC in various neurons in Drosophila. This I have to say that this type of design is a little bit, I have more circumspect about this because we overexpress DMYC and we clearly see disruption of circadian behavior of the flies shown in these examples down here. The question here is, what really is the mechanism? Are we disturbing the development of these flies? And there may be some evidence for that, because if you look at some of the pathways in the neurons, they don't look very normal. So the interpretation of these experiments is a little bit more problematic, if you will. So we don't know that we're just creating a pathologic state of these cells that cause, because they are involved in the circadian behavior, that you're just disrupting it. Nonetheless, this leads me to the next story, which is that we asked the question whether, because clock and BMAL can bind to E-boxes, whether oncogenic MYC, which occurs at very high levels, through mass action could disrupt the clock. And remember, the clock and BMAL drive circadian transcription of its target genes, largely metabolic genes, but it also drives the transcription of negative feedback loops, proteins shown here. So because the consensus site for MYC and BMAL are very similar, we hypothesized that when you have oncogenic MYC, then MYC and MAX can bind to E-boxes that are normally will be regulated by clock and BMAL. And in this case, we hypothesize it can upregulate all the negative regulators and then suppress the clock, and allowing for MYC and MAX to bind to target genes, metabolic target genes, to drive high levels of metabolism. So that's kind of the simple model, and it's work by Brian Ullman and NHA. And we first used this reporter cell line to ask whether this occurs in the cells. So this is a workhorse cell line, which has a BMAL promoter driving luciferase. We use a luminometer that measures luminescence every 10 minutes. This allows for us, after synchronizing the cells with dexamethasone or serum shock, allowing for the population of cells to oscillate in luminescence at very precisely every 24 hours. And we collect data every 10 minutes. So when I show you the data, you'll see that these are all very smooth curves. And then what we've done is to engineer into these cells an inducible MYC system, where this is a MYC fusion to a estrogen receptor hormone binding domain, where we can activate the protein by adding 4-hydroxytamoxifen, which allows for the transcription factor to translocate into the nucleus, bind to wherever it's binding to, and then confer whatever phenotype we measure. So if you look at the reporter cell line, what you can see in the control in the blue is that the reporter oscillates very nicely every 24 hours when MYC was turned off. But then when we turn on wild type MYC on in the dashed blue line, you can see that this oscillation is very dampened as compared to a MYC mutant that we also engineer into these cells separately. And this is a MYC mutant that I identified many years ago as a postdoc as a transcriptionally mutant MYC, where it has very low transcriptional activity. And in the case of the control, you can see it does not disrupt the clock when we activate the mutant MYC, suggesting this is transcriptionally mediated. And then we also collect RNAs at various time points, as well as protein out of these cells. And what you can see is, if you look at endogenous BMAL, that when in the MYC off state, you get a very nice oscillation of endogenous BMAL RNA every 24 hours. Then when we turn MYC on, we get this very severe suppression of BMAL expression shown below. So I won't show you all the data, but we have the evidence supports the model that MYC and MAX, in fact, activates all these negative regulators, primarily reverb alpha, that shuts off the transcription of BMAL shown on the bottom left here, because we can rescue this phenotype of suppression of BMAL by simply downregulating reverb by siRNAs, for example. So, and then I just want to illustrate what we see at how MYC actually can disrupt the circadian transcriptome and metabolome. And Above here is just a number of immunoblots to illustrate proteins in a circadian machinery. Rebirth alpha, which I mentioned before, is a target of BMAL. It's a negative transcription or negative regulator of BMAL. So because it's regulated by BMAL, you can see that when MYC was off, reverb oscillation very nicely at the protein level occurs every 24 hours. But then when we activate a MYC, where MYC directly transcriptionally activate reverb, you can see that it disrupts reverb expression shown here. This leads reverb then to suppress the transcription of BMAL, leading to a decrease in BMAL proteins shown in this time course plot. And then we also illustrate that various metabolic pathways are also disrupted, where we've previously found that MYC can directly regulate hexokinase 2, the first step in glycolysis, where a hexokinase protein is significantly increased by turning on MYC. And the result of this, if you look at the metabolome, where we collect samples from these cells at various time points subjected to NMR analysis, this is quantitative NMR analysis, and for the first time, it is observed that glucose levels intracellularly, in fact, oscillates in a circadian fashion when MYC is off. When we turn MYC on, we believe that glucose level becomes disrupted and very low, because now glucose is driven into metabolic pathways for biosynthesis. And of course, down the glycolytic pathway, glucose gets converted to lactate. If you have MYC off, it accumulates in the cells. When you turn MYC on, a lot of the lactate actually is extruded out into the media, shown on the far right here, because MYC stimulates a lactate exporter protein, allowing for the extrusion of lactate. And then we also found in unpublished data, if you look at the transcriptome by RNA-seq, you can find that many transcripts, as predicted, would be oscillatory. And then when we turn MYC on, you can see this marked disruption of the circadian transcriptome in the U2OS cells. So if we step back and put this all together in a number of cartoons here, we've been very interested in the concept of maintenance metabolism, which we believe allows the cells to take on nutrients, to have maintenance metabolism, to maintain biomass when proteins turn over, organelles turn over, to allow the cells to make ATP. And this is largely regulated by the circadian clock, as I illustrated. Now, in the MYC-induced proliferative setting, whether it is oncogenic MYC or MYC being turned on in T cells, that we believe that MYC then disrupt the circadian clock, as I illustrated in the model system. And this allows MYC to now to bind to E-boxes across the genome, many of which drives metabolism, such as the ones that are driven by BMAL. And this allows for an increase in proliferative metabolism to allow the cells to grow, to undergo biosynthesis, to grow in size, to prepare for cell division after DNA synthesis. So with this conceptual framework, we've been very interested in whether, if MYC is deregulated, it blocks the clock, drives this constitutive metabolism. Could we interfere with various pathways that is downstream of MYC to uncover metabolic vulnerabilities of cancers? So first, I'm just going to illustrate some work that we did that I published a couple of years ago in collaboration with Dean Felcher at Stanford and in de novo fatty acid metabolism. So I just want to point out that if you take mouse kidneys at various time points, and you look at the various transcript of that encodes enzymes in de novo lipid synthesis or fatty acid synthesis. So you see here citrate going to the various intermediates driven by these enzymes, ending up in palmitoyl-CoA, and then it can also be unsaturated and then conjugated to glycerol to form full various type of lipids. And if you look in the kidneys, you'll see that the first three steps in de novo lipids fatty acid synthesis, that these transcript actually oscillates in pretty good amplitude in the kidneys, shown here, ACLY, ACAC, and also fatty acid synthase. And just this paper was published a couple of years ago, so I'm just going to summarize what we found in terms of the model. supported by the evidence we gathered, is that what we found intriguingly is that MYC can actually regulate SRBP, the transcription factors involved in lipid metabolism, directly, increasing its expression. MYC can also directly activate SCAP, which is a cofactor that actually activates SRBP, allowing for SRBP, which is normally in the cytoplasm, to then to translocate into the nucleus. What we found by chromatin immunoprecipitation assays that it is SRBP that coordinates with MYC on the promoters of lipogenic genes to turn them on. And so what's the consequences of this? So if you take, Dean Felsher's lab has created a MYC-inducible system in the mouse kidneys to create a kidney cancer model. And when you turn MYC on, you can see on the right here is that if you look at the kidney itself, these are just slices of the kidney, that the kidneys get very big with all sorts of tumor cells, but it is reversible. If you turn MYC off, you can regain back the normal kidney. And when one takes cells out of these tumor samples and create cell lines, in this case, this is one such cell lines called E28, they still maintain the ability to turn on MYC on and off. In this case, if you look at the RNA levels of the first 3 steps of fatty acid synthesis, you can see that when you turn MYC on, a couple of these are very highly induced, including this steroid desaturase shown here. Now, for proof of concept, we asked whether interrupting de novo lipid synthesis actually could affect tumorigenesis. In this case, we use a compound called TOFA, inhibits the very first step, and the design of the experiment is in 3 ways. First, we allowed the tumor to increase over time without any treatment, so control. Or we can do a prevention measure where we treat the animals very early upon activation of MYC, where in the green line, you can see that no tumors form, that the tumor weight remains, is undetectable. But then you can also have a third paradigm where you allow the tumors to grow, and then you treat it after 2 weeks. And what we can see here, and this is probably very model specific, but remarkably, you can actually reverse the tumorigenesis, returning really decreasing the kidney size as shown here. So that's just one example where disruption of circadian metabolism, where it's replaced by increased MYC-driven de novo fatty acid synthesis, one can disrupt that. Now, my lab has also been very interested in 2 other key pathways, glycolysis, where we've been very interested in pyruvate conversion to lactate as a major so-called Warburg effect. And because the way we got into this story is almost over 20 years ago, we discovered that MYC regulates LDHA, and that's how our whole journey into metabolism started. So we've been very interested in targeting LDHA, and then at the bottom here, we also been very interested in targeting glutaminase, which converts glutamine to glutamate, allowing glutamate to actually contribute to the TCA cycle through alpha-ketoglutarate. So let me just spend a moment to tell you kind of the arduous task of trying to go after some of these pathways. About 10 years ago, I proposed to the National Cancer Institute to create inhibitors against LDH. And 10 years later, through a lot of work by many people at the NCI and at Vanderbilt University, we start out with screen for scaffolds, and then building upon various scaffolds, we now have nanomolar compounds that are orally available, that have been shown to be co-crystallized shown here is a tetramer of LDH, where the compound sits here right next to NAD in the catalytic cleft. And so these are the 2 main compounds that have been used by a number of laboratories. Now, I'm just going to illustrate in a collaborative study that in this pancreatic cancer model, depending on the administration regime, you can see that after 3 doses, compared to control, you can slow down the tumors shown on in green. If you add 3 additional doses here, you can see you can slow down the tumors further in this xenograft model, suggesting and providing proof of concept that intercepting a single pathway with a single enzyme, that you actually can affect the phenotype of this particular tumor. Now, what we found, I want to segue into something I refer to as a challenge into the field of cancer metabolism, which is off-tumor on-target effects, so-called side effects, if you will, that are on target. So what we know from the literature from many years ago is that mice with mutations, the hypomorphic mutations of LDHA, actually have hemolytic anemia. So they have this low level of anemia that's compensated. What we found interestingly when we first studied this in my lab in various models is that we noticed that they also have a drop in red cell count, and they have a compensation of anemia by increasing hematopoiesis by producing red cells from the spleen. So we know which animals are treated because their spleen size are larger. And this turns out to be phenocopy very nicely in a genetic model of LDHA knockout, total body knockout. When you have essentially a heterozygous knockout state, you get very mild hemolysis with a slight increase in spleen size shown below. But if you knock out LDHA more severely, whole body wise, you can see a drop in red cell count with a remarkable increase in spleen weight. So the reason why I point out this whole off-tumor on-target effects is that when one wants to target metabolism, we have to be very aware that while we're trying to hurt the tumor cells, are we hitting other cells as well? Because many of our cells use very similar metabolic pathways, as I alluded to in the beginning. So this leads me to just give you the conceptual framework, and what we're confronting in this field is that while we want to go after the tumor cells highlighted in yellow, that we also look in the tumor microenvironment where you have immune cells that either help the tumor cells to grow or immune cells that try to go after the tumor cells to kill them. And the ones that actually are toxic to the tumor cells are M1 macrophages and cytotoxic T cells or NK cells. So when we try to go after glycolysis, such as inhibiting LDH, we want to kill these cells. However, if you look at the metabolic features of these other cells, it turns out that the cells that are anti-tumor, that is the CD8 cytotoxic T cells and M1 macrophages, also use glycolysis. So the question here is that are we basically trying to kill off the tumor cell, but at the same time inhibiting the immune arm that would go after the tumor cells? So the net effect of this is nothing. We don't get a gain. So this leads me to just highlight for you some studies that we've done, where on the left, this is a MYC-inducible, very aggressive liver cancer model in collaboration with Dean Felcher. And so what we showed in my lab a number of years ago that Yang Ziyang and Zach Stein did was we were able to provide proof of concept when we were to inhibit glutaminase. We can find that with this allosteric inhibitor that we can prolong the survival of these animals. So this is a very striking endpoint, it's death. However, when we took these very potent compounds that clearly shows hemolysis in these animals, that we were unable to gain a prolonged survival of this animal. So we're very curious whether this is because we're tipping the scale in favor of the tumor by inhibiting the anti-tumor arm of the immune system. Now, there may be another reason for this, and this is just work from Masha Yunevic's lab that was published last year or so, a couple of years ago, where she took the genetic approach and knocked out glutaminase in this same MYC-inducible liver cancer model. And basically, she phenocopied what we found using a pharmacological approach, which is you can prolong the survival of animals when you inhibit or when you get rid of glutaminase. However, if you were to knock out the first step of glycolysis, N2, where I showed you previously is the target of MEK, in this case, you did not see any prolongation of survival at all, at least in this model, suggesting that they are probably more dependent on glutamine than glucose. However, when she combined both knockout of GLS and HK2, you can see that there must be some kind of codependency where now you can prolong the survival of these animals much further by blocking both glutamine metabolism and utilization of glucose. So clearly these cells can use other metabolites for their tumorigenesis, such as fatty acids and other amino acids, for example. But nonetheless, this is to illustrate a point I want to make, which is cancer metabolic flexibility, which is a challenge to target metabolism. So basically, we have ongoing studies to try to have a deeper understanding of this in terms of using various metabolic inhibitors on the scale here, where you have anti-tumor immune arm versus the immune arm that favors the tumor cells. So when you throw this perturbation into this mixture, which way will the balance go, and whether we end up finding inhibitors that would be of clinical benefit. So let me just summarize what I presented. Basically, what I touch on is that I started out by talking about maintenance metabolism, which is very dynamic, driven by the clock. I don't think there's any real contention here, because every organ that one looks at in a mouse at various time points, you get this very highly dynamic metabolic profile as well as transcriptome. And then we stumble on the fact that oncogenic MYC could disrupt the clock. And one could ask the question whether this model system that we use in the cell line really can be recapitulated in an organism. I can only tell you that because Brian Ullman has started his own lab, my philosophy is that trainees should take major projects into the lab. So I kind of allow that area of research to be, I used to allow to be a kind of a strong word. I encourage that work in Brian's lab. So Brian now have created a system where he has a MYC induced lung cancer model, where he has crossed that with a PER2 luciferase reporter mouse. And what we found so far is that if you looked at a normal lung versus lung that start to show signs of tumorigenesis, or just comparing control mice with the ones that has MYC in the lung, you just can see evidence of disruption of the clock. So this seems to happen in vivo as well. Now, whether that actually translates to the fact that disruption of the clock somehow plays a role in tumorigenesis remains to be explored. And then the third point I want to make here is that given the models that we generated, my lab has been very interested on a more translational arm, which is would deregulated metabolism downstream of oncogenes can be exploited as vulnerabilities of cancers, so that we can create small molecules that hopefully will end up helping patients. But the challenge, as I point out to you, and I would argue that this may be a naivete in the field over 10 years ago, is that we were very, very cell autonomous, cancer cell autonomous focus, is that we can kill the cancer cells, everything will be fine. But then a wake-up call is that, oh, by the way, metabolism is used by all sorts of cells. So if you have a strong compound that inhibits a certain enzyme, what happens to all the other cells in the body in terms of side effects or the immune system? So this is where the challenge remains for the field, and we're still actively investigating this. And I just want to end up by thanking individuals currently in my lab for all the work they're doing, many collaborators over the year that's been so instrumental to what we do, and particularly to Amita and John Hoganesh when he was still at Penn, because if I didn't spend some time at Penn, I would not have worked on the circadian clock at all. And of course, the funding agencies. So I want to stop there and thank you for your attention, and be glad to take questions.
Well, thank you very much. Yeah, let's just go ahead, directly ask questions. Anybody has questions? I'll start off with a question, out of ignorance. Are there point mutants, oncogenic mutations in MYC that are point mutants that don't?
Yes.
And has Brian or you thought about incorporating those in tissue-specific models of,
Yeah, so that definitely can be done. We just haven't done it because what Brian has done is kind of more activated MYC, that is the increased MYC levels. So there are naturally occurring mutants of MYC that happens to be in a site in the N-terminal end that is involved in degradation of MYC. So when you mutate these sites, MYC proteins are much more stable and the steady state level are higher. So that's a tool that's been used in the field. But we have not tested those alleles to see if they could disrupt the clock, at least in model system. So questions or suggestions? Up.
So somebody have a question?
Yeah, wait, is somebody else raising their hand?
Go ahead.
I wasn't able to see. Okay. I was gonna ask if like in cancer cells, are they able to like maintain and repair the mitochondria? Have you seen any of that?
Yeah, so there's quite a bit of work done in mitochondrial perturbation in cancer. So they certainly can repair. Their transplants also disrupt mitochondrial function as well when it's deregulated. What we found many years ago is that, at least in model system we have, is that MYC actually regulates genes that are involved in mitochondrial biogenesis, and others have shown that MYC also can perturb the mitochondrial fusion and fission in the cells, in the cancer cells.
Okay, cool. Thank you. Yeah, I was wondering because when you were saying that like the cancer cells, they actually turn away from like the mitochondrial oxidation, and they turn towards like glycolysis as their main form of energy. I was like, what's going on there? Like, do they have healthy mitochondria? But it's good to know. Thank you.
Yeah. Yeah, I just want to clarify the concept that may be out there that's most likely incorrect, which is that there's this concept that cancer cells switch on glycolysis and should switch off mitochondrial function. That is not true, because many cancers, in fact, both are turned on. So you turn on mitochondrial function for all the biosynthetic function of that organelle, particularly in nucleotide metabolism or synthesis requiring aspartate, for example. But then you also drive glycolysis to really deal with one carbon metabolism, to the folate cycle, for example. So everything's turned on, basically. Yes.
Yeah, so yeah, I'm wondering, so because my impression is that the MYC and the clock can mutually affecting each other. So what if you just disturb the clock? Will that affect MYC action?
Right. Yeah, no, a great question. So you know that there's been a number of studies that suggest that clock and BMAL can regulate MYC. And in fact, we think the model is that MYC is downstream of the clock in physiologic conditions, right? So where clock and BMAL is dominant, regulating maintenance metabolism and all the maintenance cellular activities. So you only require MYC when you need it, and you turn it off when you don't need it in a maintenance state. However, what we found is that if you have oncogenic MYC, you get this, if you will, this neomorphic deregulation of the circadian circuitry, right? So that's a pathologic state. So the physiologic state is MYC is downstream. So I agree with you that the question really is, in a MYC deregulated state, truly deregulated, could it still be regulated by the clock or not? I don't know the answer to that. Maybe not, right? However, in other oncogenic pathways, like for example, you have an activated RAS where MYC is just downstream, MYC is not deregulated other than being deregulated by KRAS. In that case, could MYC still be regulated at the circadian level? And whether regulating the clock will affect MYC in such a way that you can alter tumorigenesis. Okay. So what we're doing in my lab is, to really answer that question, is that we're taking a tumor system where we know a clock is still intact. So we're studying melanoma system. And we have very interesting phenotypes when we perturb the clock. I won't get into details, but in that model system, what we're hoping to do is actually using time-restricted feeding, right, to really, hone down, like make the clock much more robust, if you will. and to see if we actually can slow down tumorigenesis or not. So those are our studies that are being planned. So we don't know the answer yet, but I suspect the answer is yes, that we will see something.
Thank you.
Okay, other ideas or questions?
Well, I'll ask us another question if nobody else. So it seems like the prevailing wisdom is that when MYC is dysregulated, you get all these effects on circadian and metabolism. Is there any way to make MYC levels normal, but have it oscillate at the wrong time relative? It does, and what would that do? Is there any way to do that?
Yeah, there is. Well, at least in a grant that I wrote, theoretically it's doable, but we haven't been able to engineer it yet. So we were thinking of using optogenetics to have a light-driven any gene, right? You can put MYC behind it. And then you basically can take a system where you can create a system where you can get rid of endogenous MYC and replace it with this optogenetically regulated MYC, so you can turn on the wrong time, if you will. We just haven't been able to engineer it. So it's actually more complicated than I thought when we start out the project, which is one of the sub-aims of one of the grants I have. But there is, theoretically, there's a way to do it. And that would be really fascinating to see if you start to do things out of phase, what would happen.
No more questions?
All right. Thank
All
you.
right. Yeah,
Thanks.
so let's all
I
thank.
actually had one more question,
Oh, okay.
if I
Go ahead.
could. Yeah, so
Are we moving to the class after this?
Yes, yes.
Okay.
okay. But from what I understand, like the peripheral cells, their clock could be a little like, their clock operates separately from like the master clock, right? Like there's a master clock and then the peripheral cells. So does it matter in terms of tumorigenesis and anything you've seen as to like where that those peripheral clocks like are like operating independently from the master? Like is there any like part of the body or like in the organs where like it really matters if those peripheral clocks are, or those peripheral cells are operating independently from the master clock?
Yeah, I think that your question may have a couple of interweaving concepts in it. So the question is, the master clock can synchronize peripheral clocks, right, through hormonal and also parasympathetic system. However, as you know, that feeding can also shift the peripheral clocks away from what the central clock is doing, and other stress factors can do that too. And then, so sure, you can separate the two. What I don't know is whether shifting the peripheral clocks away from the central clock somehow favors tumorigenesis, right, in that particular organ. I don't know. I'm not even sure how to do the experiment, but that's a question that could potentially be asked if we can come up with how to design that. Rather, what we're doing is we're using syngeneic cell line system to begin to ask some of these questions, where we have tumor cell lines that still have a clock, which we can manipulate the clock, right? And then we put it back in an animal, in a syngeneic animals, and begin to look at that as a way of looking at peripheral clocks, if you will, and how that respond to, organismal circadian rhythm.
Thank you.
Okay, I think.
Okay, so because Chi actually has another thing, let's have the students stay on. Let's all thank him for a great seminar, first of all. And then the students stay on and everybody else can go away, and we'll have the roundtable just with the students. So we'll give them a couple minutes
Okay.
here. So this is pretty free form. The students just ask questions and
Okay.
just go ahead, take it away. We got
So
hands
yeah, to start out with, I got a lot of great questions. So now you can fire them away.
up.
Yeah, go. I guess I'm gonna have to call. So maybe on my screen, Grace.
Grace.
So I was wondering, just to clarify, I guess I have a multi-part question. One, is circadian rhythm both on an organismal and the cellular level? To understand that correctly?
Yeah, so basically, the way I think of it is this. So our individual cells have this molecular clock, right, that I described at Colloquium B-Mount. So the circuitry amazingly have evolved. So the cycle is 24 hours plus or minus whatever. It's amazing how it was rigged that way. And then organismal circadian really is partially regulated by the central clock, as I showed you, where it drives, for example, cortisol level in the morning that wakes your body up, you know, for activity. And then as night falls, we have a whole hormonal change that allows us to go to sleep, right? So that, to me, that's the organismal circadian behavior. So if you take a mouse and you knock out BMAL, right, the clock transcription factor, and then you compare that mouse to a wild type mouse and you put them into completely dark instead of light, dark, right? You just put them in completely dark. So we call this dark, dark. So the internal clock will continue to click along in a wild type mouse where the activity reflects almost what you would see with sunlight or no sunlight. So there's something endogenous that tells the animal, oh, it's sort of like daytime again, even though it's totally dark, and they become active, right? However, if you knock out the clock, the BMAL out of the animal, they start to lose that. When you put them in dark light, they lose that behavior of distinguishing between 12 hours activity and 12 hours of inactivity, right? It starts to shift. So clearly the BMAL controls the organismal behavior as well.
Because I was wondering, when you were talking about, I think it was the slide that said the arrhythmic hypomorphic, when you knock down MYC, and then you got this irregular, the flies were irregular, and that was clearly, at least on an organismal level. And then when you also knocked down MINT, then you recovered the organismal rhythm. And I was curious if that was also on the cellular level. And then also, because you had mentioned earlier that the internal clock is set somewhat to light and dark. And so it seems like the light and dark affect the circadian rhythm, but also vice versa. So is this like a two-way street?
So yeah, so maybe if I understand where you're going with this is right. So day, night, the light, in fact, the clock in Drosophila can sense light directly, not like our clock. There are the cryoproteins when initially actually is light sensitive, okay? And the cryoproteins in Drosophila, in fact, is light sensitive. Our cryoproteins have lost that sensation. Nonetheless, what happens in the experiments we showed is the following, right? So when you have total body loss of MYC function, the animals sensing light does no longer, they care about that. Their activity completely ignores that. So something happens to the endogenous clock, just like a BMAL knockout, for example, that the animals just lose that sense of day and night, regardless of light or no light.
Okay.
But the co-deletion of MINT was a trick we used to ask whether if we reverse this loss of function of MEK by losing the negative regulator of MINT, could we recover? And the answer is yes, we can recover very well. So that suggests that MYC transcriptional function somehow plays a role in circadian behavior. That's the interpretation.
Okay. And that was a competitive interaction where MINT replaces MYC.
Yes.
And if there's no
Yeah.
MYC, then the MINT combined.
Right.
But if there's no MINT, then okay.
Yeah.
Thank you.
So there have been mammalian experiments with the following, right? So many years ago, one group was able to knock MYC out completely in a fibroblast, in a rat fibroblast. So what happens? If you compare the wild type fibroblast to mutant with no MYC at all, and it doesn't express any other type of MYC, the cells decrease the proliferation rate by 5-fold. So they grow really slowly, okay, as compared to wild type. The interesting experiment is that you can recover this proliferation rate simply by concurrently knocking out MINT. Okay, so what that tells you, when you lose the transcriptional activity of MEK, MINT plays a negative role and shut off genes that would be turned on by MEK. So when you take that negative regulator out of the way, all of a sudden the cells start to behave like it's normal again, right? So if you want to look at it, it's like MEK is accelerator and MINT is the brakes. So if you lose accelerator and you lose the brakes, it evens it out, right? So at the molecular level, that's what we think is happening. Okay, so we'll move on to Elsa.
Hi. Oh, great talk. Thank you so much. I was just wondering, after hearing about all this, kind of my next instinct, at least, is to do some kind of like bulk RNA-seq, single cell RNA-seq, or other transcriptomic studies to kind of see more detail about what MYC is interacting with. But then I got to thinking, MYC is so highly expressed. And of course, in cancerous tissues, you're going to have like a lot of stuff going on genetically. There's a lot of kind of distortion there just because of the nature of what it is. So I was kind of wondering, is that an avenue you're thinking about? Is that something that can be done or will be useful with cancer tissue? I really have no experience with that.
If I understand your question, you're talking about whether we can use omics technology to get a little deeper insight into what's going on in the tumors. Is that the question? Okay, so I can segue a little bit. So basically, what we think, okay, we're planning to do some of these. So if we had another story I didn't talk about, which is we found that hypoxia could disrupt the clock. And we published this in 2018 in Cell and didn't get a chance to talk about it. But basically, after our work with MEK, we asked the same question under hypoxia when the hypoxia-inducible transcription factor is increased. It also has a partner protein. It binds to sequences that looks like an E-box. So we asked whether HIF and ARNT could bind to E-boxes and disrupt the clock. What we found is the answer is no. It's because hypoxia induces glycolysis, which then induces acidity when you make the environment more acidic. So a graduate student of mine found out that just acidity by itself can shut off the clock, and the way it does that is by shutting off mTOR, which is required for the synthesis of proteins, including clock proteins. Okay, so why did I mention all this? So if you look inside a tumor, you'll see there's a lot of heterogeneity. There'll be areas that are hypoxic, areas that are not hypoxic. So we think that even in the tumors that have a functioning clock, that you may have clock heterogeneity, if you will, within the tumors, right? So then the question is, how do you really tease this out? What experiments can you do? So we're now conceiving experiments where we can take tumors and then doing single cell RNA-seq, for example, as a start. So what can you gain from that? Well, what we can gain from that is that we can actually look at MEK levels. We can look at any components of the circadian clock. The trick here is that by looking at any component of circadian clock, can you surmise whether those cells still has a functioning clock or not? So there's some work in the literature that's doing this, right? And some work actually by John Hoganesh, where you can imagine the circadian transcriptome as a bunch of genes that oscillates. But these genes oscillate at different shifts in their phase, right? So gene A may be expressed first, gene B then will follow that. So you have to kind of a phase shift. So at any time point, all the circadian genes would have a fixed relationship to each other, right? In a vector space. So by knowing that, you can actually put a timestamp just purely by the relationship among all the circadian genes. And this is a trick that John Hoganesh and his Ron Anafi did. And they were able to use this for just fixed time point transcriptome to glean whether that sample has a clock or not. So I can imagine if the sequencing is deep enough and reliable enough that we actually can use this kind of very deep omics to begin to understand tumor heterogeneity in terms of whether the clock is functioning or not, and whether that is relevant to tumorigenesis. And this is just a hypothesis, is that I think this does play a role in, for example, therapeutic resistance. When you hit a tumor with a regimen of whatever drugs, well, you have cells that are resting, cells that are proliferating, cells that have no clock, cells that have clock. They're not going to all respond to the drugs the same way, right? So maybe some of the resistant disease are those that may have disrupted clock. Just a hypothesis or whatever. But anyway, so to answer your question in a long way, yeah, so there's a long way to really understand this using the omics.
Thank
Okay,
you.
so Hui.
Hi, Professor Dang. Thank you for the great presentation. My question is about MYC in embryonic stem cells in fetal development. So I understand that normally they have a different functionality compared to developed adults. And I wonder if at that stage there's a circadian connection, or if MYC activity there can be related to cancer in pediatric patients. Yeah.
I'll answer the second question first. So we do have some evidence that and this is a little bit skewed because neuroblastoma is a very aggressive childhood tumor, and it's driven by NMYC. It's another family member of MYC, which behaves very similar to CMYC, which is the one I've been talking about. Because if you were to knock in to the mouse NMYC into the CMYC locus, you can actually get fully developed mice. They have some neurological problems, suggests that NMYC and CMYC are very similar functionally. However, NMYC is more in neuronal tissues, and so that's why the neuroblastoma, which is from a neural crest origin. What we found in that setting is that you can take neuroblastoma cell lines that have very low NMYC, and they have a circadian clock. And if we were to ectopically induce high MYC expression, you can also disrupt the clock there. And if you take cell lines that endogenously have high NMYC, we can't detect a clock, right? So in that case, we think that in a natural setting, when you have high NMYC versus low NMYC tumors of the same type, that the high NMYC one has no clock. And in fact, you can almost predict from BMAL and clock expression on the outcome of these children as a predicting factor, is that if the clock is disrupted, they have poor outcome. But that's a correlation and not cause and effect. And then to answer your other question in terms of development and MYC, so we know that knockout of MYC is embryonically lethal. So the animals will go to about day 8.5 or something like this in the mouse. And it's not completely clear what the lethality is due to, but some people actually have tried to do more fine experiments, and it could be that there's a disruption in early hematopoiesis where the blood system, and then there's some other developmental problem where they can't thrive beyond that, right? And the only other thing that may be related to your question is, what about embryonic stem cells when they differentiate? So we know that in mouse embryonic stem cells, MYC levels tend to be higher than in differentiated tissues, right? This experiment we have not done, so I don't know what the results will be, but we also know that embryonic stem cells, mouse embryonic stem cells, doesn't have a functioning clock. The clock only starts to function when the cells start to differentiate into specific layers of tissues. Then the clock starts ticking in. We don't know whether that's MYC related or not, and we have not done the experiment where if you were to decrease MYC earlier on, can you start to trigger the clock function in the more embryonic state or not? We don't know. So, okay. All right. So we'll move to, is it Ceci?
Hi. Oh, hi, Dr. Dan. Thank you for the great talk. So I have two questions. So my first question is, you mentioned in your talk that cancer cells can use fatty acids as respiratory substrate. So I was wondering, is it like, because cancer cells require so much energy, so they tend to use whatever they have to support the cell development, or it's like cancer cells, sometimes they are kind of starved and they don't have enough access to glucose, so they have no other choice but to use fatty acids for energy supply. Well, I'm asking this because I'm also interested in fatty acids as respiratory substrates, although in a different organism. So I wanted to see how this works in cancer cells. Yeah.
So you're absolutely right. So I think one of the conceptual framework that's emerging is that, tumor cells to survive obviously have to rewire themselves to adapt to whatever environment, right? So at least there are a number of studies that showed, I think pretty nicely, that the type of nutrients could be cancer type specific. So for example, in breast cancer, where the tumor cells are around a lot of fat cells, okay? It's been shown in model system that breast cancers tend to use fatty acids for oxidation. So if you interrupt fatty acid oxidation, you actually can diminish tumor formation in breast cancer. And so there are a lot of interest now that whether adipocytes around a tumor cells can provide the tumor cells with fatty acids for metabolism, right? So that's an emergence. But if you look at another tumor type, that may not be the case at all. Like if you look at the leukemias or lymphomas, they use a lot of glucose and glutamine, okay, and not fatty acids. So I think it's going to be kind of tissue type specific. And the breast cancer is probably the best example for fatty acid oxidation.
Okay, my second question is very general on cancer metabolism. So I learned about Warburg effect a few years ago. It says cancer cell actually tends to use glycolysis instead of aerobic respiration for energy supply. And glycolysis is actually less efficient compared to aerobic respiration. So while that doesn't make much sense to me, so I would like to know your opinions on the controversy over Warburg effect.
Yeah. I think it's probably much less controversy than there's misunderstanding, right? So let me just, I didn't introduce it, but let me just go back a little bit, right? So basically what I didn't underscore, which I now that you asked that question, maybe the next time I give a similar lecture, I'll make it clear. So if you look in proliferative metabolism, right? Generally, I think the model is the following, is that you do have this high flux of nutrients into the cells, right? As I indicated, you have glucose coming in, making lactate, that's a Warburg effect. We also demonstrate in isotope tracing is that you can take the same cancer cell system and they use glycolysis and glutamine at the same time by oxidative metabolism. So you can have glycolytic metabolism and oxidative metabolism. They're not mutually exclusive, okay? So that's the first conceptual issue that's out there that people said, well, they're using glycolysis, so therefore they're disadvantaged. because that's the first comment, is that two things can go on at the same time, okay? The other thing that people don't talk about is that if you're only talking about ATP, right, where, you know, 1 glucose or 2 lactate gives you a handful of ATP, but if you go through the TCA cycle, you get 36 ATPs, okay? So that's the question you're asking. However, if you look at the flux, the actual flux, the rate of fluxing through the system, glycolysis is much higher flux than TCA cycle. So I can run the glycolytic pathway 50 times faster than I can run the TCA cycle, right?
Oh.
So in that case, I can generate just the same amount of ATP. It's just less efficient, right? But the cancer cells doesn't care, because as long as you have glucose, it doesn't matter whether it's efficient or inefficient, you just get what you need, right? So that's the other thing that we do know, is that the flux to glycolysis is much higher by metabolic tracing experiments in a time course fashion. So that's another piece of that also. And then the third thing I want to say is that fluxing through glycolysis is not just to make ATP, because as I showed, on branching out of glycolysis, you can make serine and glycine to make nucleotides, right? You can spin out ribose to make nucleic acids. You can spin on the other side of just the way I do my math, is you can spin out, for example, glucosamine from glucose to make carbohydrates, right? So the glycolytic pathway is not just churning out lactate, but some of it gets siphoned off to the side to make macromolecules in the growing cells. And this is pretty well established if you do tracing studies.
Oh, I see.
So it's not just ATP production, but also for macromolecular synthesis.
Okay, that makes sense. Well, thank you so much.
Okay, great. Great question. All right, now we move to Liz.
Yes. I have a small question. Well, thank you so much for the talk. It was really good. I have a small question in something that I saw on the paper. It's just a little clarification. So on the paper, you mentioned that you use 3 different drivers to overexpress the MYC, but then you also use the system, the GAL4 system. So I just want a little clarification. Are the 3 drivers in the system, or did you use the system and then you added the 3 drivers differently? And also, where in the system exactly did you put the drivers?
Okay, so as you know, the GAL4 is just a driver for any kind of transgene you want to put in, right? So then it's driven by the GAL4 site in front of your gene of interest. So what we decided to do in the paper, that's why I think the MYC overexpressing experiments are difficult to interpret, right? Because we have to express it somewhere. So what we did, not shown in the paper, I don't think we reported it. Maybe we did, I'm not sure. But first experiment we did was we tried to drive MYC expression across the entire body, right? A whole Drosophila. So what we did is we used an actin-driven heat shock protocol to activate MYC throughout the entire body. What we found with that is that the heat shock itself screw up circadian behavior. So we couldn't use it. You know, we have very nice induction of MYC protein levels in that system, but if you want to study circadian, at least in our hands, we couldn't, that just didn't work at all. So we abandoned that. And then we say, well, there isn't a great way to drive MYC expression. So why not ask the question, let's see if we target MYC in neurons, right, in cells that express specific components of the clock. And that's why you see the cry and the PDF, which is driving the PDF neurons. So there, what we're doing really, if you step back and look at it, we are just driving high MYC expression in neurons that regulate circadian clock, right? So then you can ask, well, you basically are just perturbing the metabolism of these neurons. So of course you're going to disrupt the clock and you're going to disrupt circadian behavior. So that's why I say the interpretation of the experiment is a little bit more circumspect in the sense that could we just be screwing up these neurons? That's it, their function. And if they're in a circuitry that can regulate circadian behavior, then of course you're going to disrupt circadian behavior. I think the loss of function one is a lot more telling because it's a little bit more natural, and the fact we can rescue it, right?
Thank you so much for explaining that.
Okay. Okay, so we go to C1.
Hi, Dr. Dan. Thanks for this talk. I have two questions. The first is, it's about circadian clock. So do you think there might be an epigenetic mechanism that mainly controls the resistance of the internal clock to external environmental synchronizers like the zeitgebers, environmental signals like light or so? And I mean, on the other hand, it is just the efficiency to synchronize. Is there any mechanism that can control this? I'm not talking about the sensitivity of the neurons to outside signals. I was just talking about like more molecular on the molecular level.
If I understand your question correctly, is that where does the robustness of the clock comes in? I mean, you can perturb it and you take away the perturbation and it comes right back, right? That's so what, so yeah. So so this is kind of my viewpoint, is that over time when the system evolved, right, it's just the system is rigged so well with all these feedback loops that it can reset itself, right, completely reset itself. And it's a circuitry that's established in such a way that you can screw around with it, and then if you take away the perturbation, it just goes right back. And so I think the robustness of this is really designed in the whole system in terms of transcript, transcription, transcript half-lives, protein half-lives, all the post-translational modification that has been evolved over time. So it becomes a system that can just rebuild itself, reboot itself, right? And it's just the way that it's evolved. And I don't know there's another way to explain it other than that that's the way the system is designed. And if you even if you get rid of a component of it, when that component comes back, naturally, it just kicks the system back in again. Now, I'm trying to think of a parallel out in the real world where this is the case, but I think it's just a design of the system.
I really like the expression of the half-life, the composites of the life system. But I think another level of robustness I'm curious about is that it might not just go back directly to its original state. How about the shift of this circadian clock?
Yeah. So you're getting into something it's a little bit interesting that Jerry and I had talked when we met before, is that so we have an experimental system in a melanoma system where the cells surprisingly have a very intact clock, okay? And so we chose to study it because we said, well, this is interesting. We have a tumor system in a mouse system, a syngeneic system that we can study. We can ask questions like, if we disrupt the clock, what happens? Or if we ectopically express various components, what happens? What we found interestingly in this system is that when you perturb the clock strong enough and permanently enough, that you actually can shift the state of the cells, okay? So basically in this system, which we're still working on, I didn't have time to mention it, but basically we can take a cell, a melanoma cell that's relatively well differentiated, and if we ectopically express a dominant negative BMAL in it, we actually make the cells more undifferentiated. So we now disrupt the clock. They can't recover because the dominant negative continuously going to disrupt the clock, right? Then all of a sudden the cell itself, cell state change. So that I find it really fascinating that maybe what the functioning clock is doing in systems like this is to define the cell state, right? So for example, if you look in your tissues like the colon, where you have various very distinct cell types in the intestinal crypts, well, why do they maintain that transcriptome? how do they maintain that identity, right? In addition to external signals. My own belief and hypothesis, we're going to test all this out, is that the clock actually causes a stability by just causing an oscillatory system that remains in that state, right? So if you're into this whole area of non-equilibrium thermodynamics, which people talk a lot about, oscillatory state that's very stable, like you can make them unstable and they reach a different oscillatory state, right? So I think maybe different types of cells have a clock to maintain the state they're in.
And like, so my second question is regarding the mechanism, are you like mostly believe that's going to be transcriptional regulation that might be the major contributor?
Yeah, I didn't catch the last part. What is a major contributor?
Oh, I was asking, do you think like transcriptional regulation is the major contributor to the mechanism to either stabilize or synchronize to?
Yeah, I think it's everything together, not just transcriptional, right? Because you know that there's an arm of the clock that is the dominant arm of the clock is actually non-transcriptional through cry and purrs, right? So I think I mean, the system has evolved to be robust to have so many feedback loops. And, you know, in that case, we don't, you know, the major components have been found. I would argue, however, from the work that we did in the Drosophila with MYC knockout, is that could there be other minor components of this that we don't understand? When I say minor, is that the phenotype is not shown, but you saw how loss of MYC and Drosophila screw things up so much that you can recover by double knockout, right? So of MYC. So whether there are other so you know, one way to look at biology is this, right? I mean, for the cancer fields, that I think we, over the last 30 years, we know a lot of the major hubs, right? So let's say clock and BMAL is a major hub for clock, but how many other little things around it that keeps it that robust, we don't know. I don't think we completely understand all of it. It's the same thing with all the oncogenes and tumor suppressors. We've discovered most of them, not all of them, but most of them. But what we're dealing with is even we know that. Why is it that a patient with similar mutations responds so differently to the same drug? Because of all these other minor factors that modifies the hubs. And that's where, you know, I think that's where the new biology is going to come in.
Thanks. That's just really interesting.
Yeah.
I guess the ultimate question is going to be which part of this mechanism are, you know, inheritable, like in terms of transmission genetics, but I would leave that question to the future. Thanks.
Yeah. Okay. Great. And then Ryan.
Hello. Thank you for the talk today. It was great. My question's probably pretty simple, but like, do you know how like MYC responds with like treatment of rapamycin, or like if overexpression of MYC like negates like translation inhibitors or like analogs of rapamycin or something?
Yeah. That actually is not a simple question. It actually is a very good question. And there's some evidence in the literature that MYC protein's translation can be downstream of mTOR. So it's almost like a symbiotic thing between TOR and MYC, right? I show you the simple model, stimulate cells to grow. TOR gets activated separately by amino acids and growth factors. MYC is stimulated downstream of ERK and the protein levels. But if you inhibit mTOR, MYC protein levels cannot be made, right? So they have crosstalk. And then there's some evidence, although they're not, I would argue not strong enough, but it appears that in one system that MYC could be a negative feedback loop for the TOR pathway, because when you activate MYC, it actually increased TSC2, okay, which is an inhibitor of the TOR pathway, right? So I think we have unknown circuitries that have not been mapped yet, but I do know that if you treat with rapamycin in a certain system, MYC protein levels go down. So that we do know.
Okay, cool. Thanks.
Okay. And then I think we have Angelica.
Hi, that was a really nice presentation. Thank you. My question is a little bit off topic, I guess, but I was just really curious a bit about your career path since you kind of went PhD in chemistry to MD to now doing research on cancer. And I was just curious what kind of led you down those different paths and how you kind of settled on your current career.
Yeah. So, thanks for the question, but I can share very briefly, right? So I have to say, I love science and I love almost everything that I do. So I never really planned my career. I can tell you that. When I started out, I just enjoy the courses. When I was in college, I love philosophy, for example, and then you meet some professor that really excites you and you start doing that. And sort of that's kind of where I was going, right? So the angle on the medical school part is that I do have a little bit of familial influence because my father was a physician. So there was always in the back of my mind is that, do I want to be a scientist or a physician scientist or what? So I ended up going to medical school after I got my PhD. But again, I headed right back into research as the major part of my career, right? And so I never really planned it. I just like one step at a time. And, you get, like I said, good timing and luck has really been my friend along the way. And, I even venture into doing administration and various jobs, it's just whatever is exciting. But one message I do have for career going in the future is do stay some thematic focus on what you work on, right? Because you can jump around different topics. You never can get too deep into an area that you understand and enjoy deep. So I've stayed with MEC for many years and it's led us into all sorts of very interesting stuff. We're now doing more immunology because MEC is also implicated in immunologic functions. So we're now, half of my lab is doing immunology now, right? We never plan on it. So I don't plan on things. I just go in part where our interest goes and things like that. So that's the answer.
Interesting, thank you.
Okay, Megan.
Hi, yeah, so I had a question. You had mentioned like earlier in your talk about like the differences between Drosophila and the mammalian like MYC genes. And you mentioned that they were pretty different, but like still acted by similar mechanisms. And I was just wondering, like, I know plants also have a circadian rhythm. I was wondering, like, is it thought that these clock pathways all evolved independently, or is there like some similarity between the clock genes in different organisms?
Okay, so maybe I can answer the first part. So you asked the question of whether the difference between mammalian MYC and Drosophila MYC, right? So
yeah.
So there are a number of experiments other people have done. We didn't do this, right? So if you take a Drosophila MYC mutant, you can actually partially rescue it with human MYC, right? So what that means to me is that there's some conserved function between MYC proteins of very diverse organisms. So those experiments have been done. But however, clearly the components that binds to MYC in Drosophila are actually, they're not the same. They're quite different than all the components been identified for human c-MYC. So I would say that the actual function of DMYC in Drosophila is probably different in some special way, but the commonality which has been established is that what we found in mammalian system is that MYC can turn on metabolism because many metabolic genes have E boxes. Most metabolic genes in many cells, the core metabolic pathways have open chromatin. So when MYC is high, it binds and it turns all these genes in. The same thing has been found in Drosophila, so I think that's conserved. The other area that's very clear is that Drosophila MYC regulates ribosomal biogenesis. Many genes in Drosophila that's involved in generating ribosomes are MYC target genes, and you can phenocopy the ribosomal biogenesis phenotype with MYC mutations, right? The same thing is found in mammalian system, which is one of the prominent group of genes that's regulated by MYC in almost on all system is ribosome biogenesis. So that's conserved. I'm absolutely sure there are differences. I just don't know what those things are. So we do know that there are MYC targets that are cell type specific. That happens. There's no doubt. We've done this, right? For example, in the extreme case, if you take human embryonic stem cells and you take a differentiated human cancer cell line and you do chip analysis to look for MYC-bound genes, and then you look at the transcriptome for MYC-responsive genes, there are clearly genes in embryonic stem cells that are MYC responsive because we knock down MYC, the gene expression goes down, it chips there. None of those genes are expressed in the differentiated cells because chromatin have been shut down, for example, right? So I think it has a lot to do with chromatin accessibility for the MYC-MAX transcription factor.
Okay, that's interesting. So do you know if MYC still binds in those differentiated cells, or is it not even able to bind?
So where the chromatin are shut down, you can no longer access it, right? But the differentiated cells, in many systems, MYC expression is very low. So whether MYC plays any role there is unclear. Now, we've done one experiment where we've knocked out MYC in mouse liver, and then we asked the question whether MYC is required at all for liver regeneration, right? So we would cut a big portion of the liver and then it regenerates. For some reason in the mouse liver, it does not depend on c-MYC at all. They can still replace. So again, is it possible that the transcriptome that's responsive to MYC, as long as the chromatin is open and of some other transcription factor can also regulate it, that it can replace MYC, right? So that's another complexity of that's worth mentioning.
Okay, that's very interesting. Thanks.
All right, Manuel.
Hi, thank you for the great talk. It was very entertaining. I was wondering, regarding the different types of cancers, I know there's some that they have abnormal MYC expression and some of them don't. And I was wondering, like, how does that relate to the circadian rhythm?
Yeah.
In terms of like, what are the cancers that have normal MYC, and does that mean that they also have a normal circadian rhythm yet are cancerous?
Yeah, no, that's an excellent question with no clear answer yet. Okay, so I can only share with you what's available in the literature, and what we do know from my lab is that I mentioned before, there was an algorithm that's called Cyclops, created by Ron Anafee and John Hoganesh, where by looking at a single time point transcriptome, looking at relationships of the circadian clock and target genes, that you can put a timestamp on the samples, okay? So even it's just a snapshot, you can say, this sample is likely to have a functioning clock, and this is the time of that clock according to that transcriptome, okay? So what they did is they took many samples of liver cancer versus normal liver, and these are all single time points, right? And when they start to use this algorithm, they actually can look at all the normal tissues, which happens to be sampled at different time of day. They can put a timestamp on when the samples are taken from just the transcriptome. And you can see very nicely in the normal liver that there is clearly a circadian behavior to the samples, okay? They did the same thing with liver cancer, and you can't see this pattern. I mean, basically, you can't put a timestamp on them. They kind of scattered all over the place. So it seems the interpretation is that liver cancer have disrupted clot. It turns out that at least in human liver cancer, MYC tends to be pretty deregulated, but that's just a correlation. It's not, we don't know if it's causal, okay? Now, our current study in my lab, we're looking at a murine melanoma system that has been generated through transgenic mice that have other oncogenes, not MYC, right? So it's BRAF mutations, it's beta-catenin and INK4A. So these are non-MYC oncogenes that drives melanoma formation in the mouse. So we've studied the cells that have been made out of this, and you can put it back into the mice. It turns out these cells have a completely functioning clot, and they express very low MYC, right? If you overexpress MYC in these cells to disrupt the clot, what it doesn't tell us whether, to answer your question, is that are all tumors of high MYC have no clot, and the one that have low MYC still has a clot? We don't know. We just don't have enough data to really answer that question. But those are the two anecdotal things I can tell you about.
Thank you.
Okay, so I don't see other hands up that we could.
I had a question.
Yeah, sure.
Yeah. So first of all, great talk. Thank you. But I had a question regarding the heterogeneity of cancer. So of course, any cancer, like tumor that you're going to look at, is a heterogeneous mixture of cells. So within that, there'll be some that are highly expressing MYC and some that are lowly expressing MYC. In vivo, would you expect that a tumor would have a different circadian rhythm to surrounding tissue? And I guess like the second part of that would be like within the tumor itself, do you expect highly expressing c-MYC cells to kind of outcompete the lowly expressing cells, and therefore the tumor itself would have a different circadian rhythm?
Right. Yeah, so to answer your first question about heterogeneity and clock function and MYC, right? So we don't know the answer to that, but there is an experimental system that I'm exploring to import, right? So people have now made animals where reporter of the clock using luciferase, like a PER2 driving, and where you can look at single cell, right, for their clocks. So one can imagine that if you take that system, you might be able to study at single cell level whether there's heterogeneity of the clock and whether the heterogeneity of the clock is related to MYC levels or not, right? So that's something that could be done. I don't know the answer to that, whether that's going to be the case or not. But to answer your second question about competition, that in fact, it is known pretty well that MYC, Drosophila MYC, is a model for cell competition, where if you take cells that have high DMYC, Drosophila MYC, next to a cell that have low Drosophila MYC, the high MYC actually outcompetes the low MYC expressing cell and actually kills them. It's not just outcompeting them. And the mechanism is really still not completely defined, but it's really fascinating that MYC does confer an advantage for cell competition. And it's been implied it could be metabolic. If you're much more metabolically active, you're going to be robbing all the nutrients from the surrounding cells. But I don't think it's as simple as that. That could be part of it. So that's kind of the second part of your answer, is that, yeah, so there's evidence that's clear evidence that cell competition can be driven by MYC.
Okay, thank you.
All right. So we have Akihiro.
Hi, thank you so much for your great presentation. I'm not actually a student, but I want to ask the question. So, so you mentioned about that MYC is a downstream of mTOR. So I'm kind of wondering the role of the MYC in the aging or age-related kind of symptoms. So have you looked at the, for example, lifespan of your mutant flies or?
Yeah, so we haven't looked at this, but there's a group that published a pretty nice paper probably about 5 years ago, where they did an experiment where nobody wants to do. I don't know how many graduate students who want to do it, but basically, John Sedevy, what he did was he compared littermate wild type mice and heterozygous MIC.
Okay, in the mouse. Oh, okay.
In the mouse. And so they bred this, they followed these animals for like well over a year, right? And what they found interestingly is that the MIC heterozygous with one copy, they live longer than wild type. Okay. And so it suggests, and their paper didn't show mechanism, but they suggest, they look at the transcriptome, they suggest that it's possible it's lower metabolism, right? Mitochondrial function and ribosomal biogenesis that allows these cells, I mean, these animals to live longer, just like the mTOR story, right? Yeah. So again, that's the only paper out there that actually have done it very nicely in the mouse. Yeah. So that's,
did you look at the lifespan in your flies?
I'm sorry?
Did you look at your flies lifespan?
No. So Bob Eisenman did look at that. So the flies that have lost MIC, actually the flies that lost mint, right, where MIC is higher, right? They live shorter. That I know. The flies that lack MIC, I can't remember whether they live longer, but I know the flies that loses mint, where you have unantagonized MIC, they live shorter.
Okay.
So too much MIC activity makes them shorter.
Okay. Thank you.
Yeah. Okay. So I think I saw Myron before Manuel. So Myron.
Hey, thanks for the talk. I had, I was just sort of interested in when you're looking at the arrhythmic responses. It was maybe naively interesting to me that you see like a fairly big difference in like variability in the behavior. Some of them you some of them you didn't even get a strong response to like the DMIC induction. Do you have any are there any insights on? Do you think that's just the inefficiencies of trying to induce DMIC expression, or is it is there something interesting in that differential response?
Yeah, so we don't quite understand exactly why the diversity of phenotypes we saw, right? What we do know, and it's very hard to say, we try to look at whether MIC levels were different among these guys, but it's whole head, right? And you can't distinguish from that. So I think the interpretation I have is there's some stochasticity in the system we don't quite understand. It's not about total MIC levels. We don't know about it at the cellular level. We didn't stain for it, but at least you look at total MIC, that doesn't seem to explain it. So we think that there's some stochasticity that maybe the amount of MIC we have is just at a point where you can get divergence of phenotype, right? Just enough to see some that are very disrupted, but not quite enough in others. Just you at maybe at a branch point where you can where you can have, more stochastic outcome of the phenotype. So imagine if we were able to express MIC even higher, could we really push the phenotype more? I don't know the answer to that. So that's the best guess.
Yeah. Okay. Thanks.
Okay. And then Manuel, I think you have a question. At least a hand's up. Okay. So any other questions?
No, we should let you go because you have another appointment. I know.
I think I'm all good, and these are really great questions. Thank you so much for spending the time, and I hope you can continue to enjoy the class.
Yeah. Well, thank you.
Thank you so
Great
much.
talk.
Yeah.
Very good research. Thank you.
My pleasure. Bye-bye.
Bye-bye. See you.
Thank you.